Thank you very much for the kind introduction, and thank you for giving me this opportunity to present our work. Today I'm going to present our work, which is to study the association between COVID-19 and changes in brain structure in UK Biobank. And this work has been published in Nature recently. And as you can see here, it is the result of many people's hard work. And I'm happy that I'm given this opportunity to present our work at this webinar. So to give you some background information on COVID-19 and its effects on the brain, there is strong evidence for brain-related pathologies. There are many papers reporting that COVID-19 patients experience neurological and cognitive deficits. And one consistent clinical feature is the disturbance in olfactory and gustation in the COVID-19 patients. And a recent study, for example, reported that 100% of the patients in the subacute stage of the disease displayed signs of reduced ability to taste, and 86% of the patients showed either hyposmia or anosmia. And some of these neurological deficits could be a consequence of viral neurotropism or of virus-induced neuroinflammation. And radiological and post-mortem tissue analysis demonstrated the impact of COVID-19 on the brain, and several studies found evidence supporting possible the presence of the virus in the central nervous system. If we focus on brain imaging studies related to COVID-19, most of them to date have focused on acute cases and radiological reports of single cases on CT, PET, or MRI scans. And several abnormalities such as white matter hyperintensities and hypoperfusion have been reported. In brain imaging so far, from what we know, have all scanned participants post-infection. So the participants were already infected by coronavirus and then were scanned. So one of the limitations of these post-infection scans is that it remains unknown whether any of these abnormalities predates the infection of COVID-19. And that's where UK Biobank imaging study comes in, as it offers a unique resource to overcome the limitation. So for those of you who may not know about UK Biobank, it's a long-term prospective epidemiological study that has many different kinds of information, such as genetics, blood samples, lifestyle information, such as whether you have eaten apple yesterday, and composes of 500,000 UK participants aged between 40 and 69. And all this information on the 500,000 participants were gathered back in 2006 to 2010. And basically people were asked via general practitioner whether they want to join the study. And although UK Biobank participants were drawn from a general population, we have a little bias because these people are slightly wealthier than general population and slightly wider. They are also slightly healthier to go to see, their GP regularly, and they want to take part in research, taking their time to participate in research. So in 2014, the UK Biobank imaging study began with the aim of re-inviting 100,000 participants of the 500,000 participants to undergo brain and cardiac or abdominal MRI scans, X-ray, and etc. And it was 2016 when we had just passed the bar of 10,000 participants who did brain scan. And we pre-processed 5,000 subjects, participants of them, and more were available later on. And by the end of 2019, we reached above 40,000 participants. Yes, and the brain imaging scans were collected from 3 dedicated imaging centers in the UK. And we also had started re-imaging study, bringing back 10,000 of them, but this came to hold due to the pandemic. And then Stephen Carla, who designed the UK Biobank imaging study protocol, said that maybe it would be helpful to have some of the participants come back who have had COVID-19. And that's how COVID-19 re-imaging studies started in February 2021. And with that said, the participants in this study is biased in some sense. So people were recovered well enough, at least to want to spend entire day doing the full body scan, including brain imaging scan, and answering questions, filling out questionnaires, etc. So obviously we are looking at much more mild to moderate COVID. And these participants who have had COVID, and I will explain this in greater detail later, were paired with one-to-one, so paired with controls. So the whole idea of this study is to investigate longitudinal effects associated with the infection. So participants in both groups were already scanned once before the pandemic, and they were scanned on average 3 years later, and the case group were infected in between the 2 scans. And what this gives you really is a really crucial baseline scan before infection, which makes it easier to disentangle the contribution of pathogenic process from pre-existing differences in the brain or certain risk factors of the participants who are in the future infected by COVID-19. So as I was saying, I will talk a bit more about study participants. So their COVID-19 status was determined on the basis of different kinds of information. One source we used to confirm their COVID status was their health records. We had linkage to their health records on the GP and hospital records. So whenever people started to feel sick or knew that they were in contact with someone who caught COVID-19, they went and did a PCR test. And another source was antibody test results. So UK Biobank sent out some antibody, not antigen, lateral flow test kits. And they were, if the participants returned positive results, then UK Biobank sent a second test kit from a different company to confirm the status. And negative status was based on either no COVID-19 test results at all on all health records or negative antibody lateral flow test. And at the end, we ended up with 401 participants with usable good quality scans at 2 time points, which is before and after infection. And we had 384 for controls. And as I briefly mentioned, the cases and controls were paired one-to-one initially, but however, you can see that we were already losing some of the participants from pairing because the fact that some of them did not have good quality MRI scans at both time points. So we're now saying that the groups are not paired, but they're matched for age, obviously age at each of the 2 scan time points, sex, interval between the 2 scans, and ethnicity. And for ethnicity, we didn't have the power to do like fine grain differentiation. So what we did with the participants was white versus non-white. And we also took into account the first imaging assessment location because we might find some slight local differences due to environment and scanner itself. You never know. So the case and control groups were matched for all these variables. And a bit more detail on the participants. So despite the original pairing of the cases and controls, the age distributions were slightly, but not significantly different due to the different patterns of missing data. So for example, here, you can see that the control group on average tended to be slightly older than the case group, but it was not significantly, I mean, the 2 groups were not different. And this difference would make actually harder to detect what we have found in our study. I mean, the fact the control group is slightly being older. And we checked for usual suspects such as high risk factors, blood pressure, high blood pressure, for example, diabetes, weight, BMI, alcohol intake frequency, smoking, etc. And all of these were based on last observation carried forward because UK Biobank has several instances of visits. So these values in the table were measured at several different time points, and we took the values that were closest to the baseline scan. And we also explored a whole array of other non-imaging phenotypes, approximately 6,300 variables, and the 2 groups showed no statistical differences across all non-imaging phenotypes after FDR or family-wise error correction for multiple comparisons. So focusing a little bit more on the positive cases, what we see is that what we see here is that we got a breakdown of the participants based on whether the information about the positive status comes from medical records such as GP and hospital or antibody home-based lateral flow kits. And the 651 participants, the information came from GP and hospital records as well as diagnostic antigen test, and the rest were coming from antibody home-based lateral flow kit. So for these 50 participants, unfortunately, we don't have a clear window when these people had been infected, but the rest people, we have that information. And we had 15 hospitalized participants who had been very unwell and hospitalized with COVID-19 as primary cause or secondary cause. And as I mentioned before, these COVID-19 imaging study were based on voluntary recruitment. So it is highly likely that if they had not been recovered well enough, they would not be very keen on participating in the study and then spend the entire day doing the whole body imaging scan. So in summary, our infected participants were very much geared towards mild to moderate cases. And looking at the time of infection closely, we can see a small peak around April 2020 and a larger peak through October to December 2020, which represent the first and second waves of COVID-19 in the UK. And based on this infection date distribution, it is likely that a small minority of the participants were infected with the original strain, and a majority of them with the variants present in the UK from October 2020 onwards, which is predominantly alpha, but also beta and gamma variants. And presumably very few participants, if any, were infected with the Delta variant, which only appeared in the UK in April 2021. And the histogram on the right-hand side shows the distribution of the time between diagnosis and second scan. And more than 80% of the participants had less than 6 months between infection and their second scan. So what do we do with the brain imaging scans? What we have done in the past with UK Biobank brain imaging data is to try to extract and condense the information. So the brain imaging protocol includes 3 structural scans, the T1, T2 FLAIR, and susceptibility weighted MRI, as well as diffusion MRI, and resting state functional MRI. So T1 scans give us global measures of the brain and CSF volumes, as well as localized measure of gray matter volume and cortical thickness and the area. And T2 FLAIR scans are useful for detecting inflammation or tissue damage. And susceptibility weighted MRI is sensitive to iron and myelin content. And diffusion MRI gives us information on the tissue microstructure integrity. And resting state fMRI, from that we can get functional connectivity between resting state networks. So from all these different modalities, we get 1,524 imaging-derived phenotypes, including variables such as regional gray matter volume, local cortical surface area, white matter hyperintensity, volume, fraction anisotropy, and etc. So the details of these IDPs are described in detail in Collins' 2016 Nature Neuroscience paper. And for this study, we also generated around 1,100 IDPs using quantitative susceptibility mapping, which is an emerging MRI technique that measures tissue magnetic susceptibility and has been shown to detect pathological changes in tissue, iron content, myelin, and calcification. So in total, we have 2,630 IDPs derived from brain scans. And in this study, we took two approaches to investigate COVID-19 effects on the brain, which are hypothesis-driven approach and exploratory approach. And our main model was hypothesis-driven approach, where we chose to focus our primary analysis on a subset of 332 regions of interest that we selected a priori from the total 2,630 IDPs. And we chose these ROIs because we came from the angle that we had enough studies showing that we should focus on olfactory and gustatory regions. So we focused these subset of ROIs functionally connected to olfactory and gustatory regions. And these regions include the piriform cortex, parahippocampal gyrus, entorhinal cortex, and etc., as you can see on this slide. And here is our statistical model to investigate effects of COVID-19 on the brain. So I won't go into like too much details of this, but the way we've done this is equivalent to looking at longitudinal differences between the case group and control group. So in the model, we have IDP1 and IDP2, where the number 1 indicates the baseline scan and number 2 indicates the second scan. And IDP2 is expressed in terms of the combinations of the IDP1 and the case versus control group difference regressor and confounds. And the case versus control regressor was created by first we demeaned the binary variable and then we modulated it with the age dependent constants. And the age dependent term was taken from a meta regression analysis study that looked at the age dependent infection fatality rates for COVID-19. And we know clearly from the literature that there is higher detrimental effects of COVID with age. So here we looked at objective statistical model based on the meta regression model of this age modulation of detrimental COVID-19 effect. And the confounds included the term of the age difference between the two scans and difference between the squares and ethnicity, sex variable. And with this model, basically what we are looking at is where do we have longitudinal changes in the brain different between the two groups that gets worse with age. And for the rest of my talk, I will refer to the main age modulated group comparison analysis between the two groups as model 1. Okay, and we performed secondary follow-up analysis to compare non-hospitalized cases against controls, which is model 2, and hospitalized patients against controls. And we also have analysis on hospitalized versus non-hospitalized cases, which is model 2. So for all these 4 models, the testing was done using the hypothesis-driven analysis and then the exploratory analysis. So therefore, we carried out 8 group comparison longitudinal analysis in total. So here are the results from the hypothesis-driven approach in our main model. which is model 1, where we compared all cases and all controls, we found 68 significant results after FDR correction for multiple comparisons, and 6 of them survived family-wise error correction. And the table here focuses on the top 10 most significant associations from Model 1. And 8 of these IDPs cover similar brain regions functionally connected to the primary olfactory cortex. And basically, we found a greater longitudinal increase in diffusion indices for the case group that have functional connections with the frontal and temporal piriform cortex, as well as the olfactory tubercle and anterior olfactory nucleus. The other 2 of the top 10 IDPs were the left lateral orbitofrontal cortex and parahippocampal gyrus. So here I am showing you the illustration of some of the results. So here the turquoise color represents controls and orange color represent the cases. And you can see on the left-hand side that the left hippocampal gyrus showed greater reduction of tissue intensity contrast in cases compared with controls. And you can see clearly the effect of age, that the group difference gets more dramatic with age. And you can see that it actually starts quite comparable in like 56, like the early ages, and then it gets diverse more with age. And same story for the left orbital frontal cortex, which is shown on the right. And again, same story. If we look at how much damage, microstroke damage occurring in the tissue, the left side shows the longitudinal % changes with age for the 2 groups in the oriental dispersion in the temporal piriform cortex, and the right hand side is % changes in isotropic volume fraction of the olfactory tubercle functional network. And these 2 diffusion indices increase with microstructural damage, and you can see very clearly the divergence that gets worse, that gets greater with age between cases and controls. So these are the main results showing longitudinal differences between the 2 groups based on our hypothesis-driven approach, where we selected the subset of the ROIs. And for the significant IDPs, we found on average, we found the average percentage change differences between the 2 groups was moderate. It ranged from 0.2 to 2%, and the largest differences were found in the volume of the parahippocampal gyrus and entorhinal cortex. And as a kind of like a sanity check, we looked at all labels covering the entire brain, including cerebellum and every part of the cortex in this exploratory approach. And from the main model, where we compared all cases and controls, we found 65 significant longitudinal differences, and 5 of them survived family-wise error correction. And what was really interesting was that half of these significant IDPs overlapped with the brain regions that were pre-selected a priori in our hypothesis-driven approach. So this really tells you that there is clear, focused results. In other words, the localization of the results toward the olfactory, limbic, gustatory region of the brain. Obviously, what this exploratory approach gives you is to look outside of the regions to answer the question whether there's any other differences in the brain, and indeed there were. So something that came up at the top, some of them were really like most significant results. It was some global atrophy. So you can see in this table, the top results included the ratio of the size of the brain compared to the volume inside the skull. So it's kind of like a normalization measure, or increases in the size of the ventricle, CSF, and decrease in the brain volume without ventricles. So this is something that really speaks the evidence that there is some local reduction in the cortical thickness and the volume of gray matter regions that are very focalized, but there are also some global changes in the brain. And in addition, in this table, I draw your attention to the fact that we've got very clear difference here in the cerebellum cross-2, which is a cognitive part of the cerebellum that is relatively expanded in human compared to non-human primates. It's very cognitive area and also very olfactory as well. And here I'm showing you the illustrations of the global brain volume changes, as well as the volume of the cerebellum cross-2. So you can see a greater volume reduction in the case group. And similar to the figures that I showed you earlier, you can see very clear divergence that gets greater with age between the two groups, and the same for the cerebellum. And this is just the visualization that shows longitudinal group differences in gray matter thickness and mean diffusivity changes. So the map here shows that the strongest localized reduction of gray matter thickness in the participants who contracted COVID-19 compared to controls, and the reasons were bilateral and parahippocampal gyrus, anterior cingulate cortex, and temporal pole, as well as in the left orbital frontal cortex, insula, and supramarginal gyrus. And when we look at microstructural damages in diffusion MRI, the strongest longitudinal differences in mean diffusivity could be similarly seen in the orbital frontal cortex and anterior cingulate cortex, and in the left insula and amygdala. So far, I have shown you the result from the main model, the model 1, where we compared all cases against the controls. And here, if we focus just model 4, where we looked at 15 hospitalized cases versus non-hospitalized cases. Well, first of all, I should mention that there's just a small number of hospitalized cases, and 2 of them received critical care. And if we compare the 2 groups, what we see here is something that is not really surprising to any of you, because it shows that the hospitalized patients were older, have higher blood pressure, higher weights, and more likely to have diabetes. So in the interest of having them in a more constrained model, we added these additional confounds in the model. So we added age differences, confounds, and blood pressure, and etc. To compare these two groups. And when we compare the two groups, so we're looking at only cases here. What we see again is very, very clear results on the cortical surface. So you can see very similar reasons such as orbital frontal cortex, perihippocampal gyrus, and anterior cingulate cortex, which were the same reasons that we found from our main model 1, where we compared all cases compared to controls. So here we see that these reasons just worse in the hospitalized cases compared to non-hospitalized cases. And we also additionally found differences in the frontal parietal and temporal cortical regions, especially on the left hemisphere. And finally, what we wanted to look at was cognition. So for this, we selected a priori 10 cognitive variables from UK Biobank that are sensitive to cognitive impairment. So what we did is was that we drew these cognitive variables from using a data-driven approach based on out-of-sample participants who are the most likely to show cognitive impairment. So specifically, we identified current and future dementia cases in UK Biobank and compared their cognitive scores with those from the matched controls. We identified 778 participants who took a comprehensive set of cognitive tests in UK Biobank. And around 2% of them already diagnosed with dementia at the initial assessment visit, cognitive test, and around 10% of them were diagnosed with dementia at the time of online cognitive test, and the remaining 89% of the participants were diagnosed after their cognitive testing. And we then matched these participants with controls, and as you can see here, that the two groups were matched very well for ages and at both initial cognitive tests and online cognitive tests, and as well as other variables such as blood pressure and socioeconomic status and etc. So the top most significant variables from this out-of-sample analysis were 3 variables from the trail making test you see here in the figure. From here, we drew we took variables that show that represent the duration to complete the numeric trail and the duration complete the alphanumeric trail, as well as the total number of errors made during the completing trail B. And we took 1 variable from numeric memory and 1 variable from symbol digit test and 1 measure from reaction time. Then we also had fluid intelligence score and we took 3 variables from pairs matching test. So in total, we had 10 variables, cognitive variables from 6 types of cognitive tests. And what we found was that the time taken to complete Trail A and Trail B were longer in the COVID-19 infected group compared to the control group, and the difference was greater with age. So this shows that there is a greater decline in the task performance in the infected group that gets more prominent in older adults. And we excluded 15 hospitalized cases, and the results remained significant. And what was interesting was that the duration to complete Trail B on the right-hand side, we found that it was correlated with the volume of the cross-tube of the cerebellum that I mentioned before. So in terms of discussion, this is the first longitudinal imaging study where COVID-19 positive participants were scanned before infection. And our study showed a significant deleterious impact associated with the infection. And that was mainly in the olfactory cortical system with the diffusion measures in the regions that are functionally connected with the piriform cortex, olfactory tubercle, and anterior olfactory nucleus. And we also found a gray matter thickness and contrast change in the parahippocampal gyrus and orbitofrontal cortex. And as I said before, these changes were very localized differences in the brain, we also found some global changes, such as the whole brain volume decreases in the infected participants, and hospitalized cases had greater loss of gray matter than non-hospitalized cases in the regions that are shown differences between cases compared to controls. We also found greater cognitive decline in the infected group compared to controls. And that was also correlated with the longitudinal changes in the volume of cerebellum. And obviously, we have several limitations in our study. The most unfortunate to us is the lack of clinical severity information, because we only have the information on hospitalized versus non-hospitalized. And another limitation is that there is small number of ethnic minority in our study. This is rooted of some bias present in the UK Biobank. And one issue inherent to the recruitment strategy is that we have very mild to moderate cases, but this can be seen, this can be considered as actually like a strength of this study, because majority of the brain imaging publications have focused on moderate to severe cases. So there was a need for more information on the mild cases. And finally, on the imaging side, we didn't have sufficient resolution to segment the whole, to segment the brainstem. So that was a bit unfortunate. So this would be my last slide. So So why the olfactory cortex? There is evidence that coronavirus itself, there is evidence that the olfactory related symptoms precedes the symptom of respiratory symptoms, and there are many reasons why we see the changes that we saw in our study. It could be due to neuroinflammation, or it could be due to sensory deprivation as well, or chronic inflammation related to nasal cavity. So we have a lot of questions regarding whether the changes are reversible, which probably I can discuss in the Q&A sessions related to this study. These studies that showed some sort of like a recovery from the brain changes in the participants who infected with COVID-19. So, okay, with that, I'd like to finish my talk, and I'd like to thank all of the collaborators from the Wellcome Center for Integrative Neuroimaging, Oxford. And the first picture here is Gwen, the first author of this paper. She wanted to join this webinar, but she had another meeting to attend, so unfortunately she couldn't join. But yeah, so I was very happy to present our work, and thank you very much for your attention.